Thanks to the organizers for the opportunity to present today. And so what I hope to really get across to you is the importance of mass spectrometry-based omics, proteomics, and metabolomics in drug development and discovery. And then also highlight, of course, for this audience, kind of really the pivotal role that computational mass spectrometry specifically plays in drug development and improving some of our traditional or existing ways of working. And so I'm coming from what is the Center for Genomics Research, or CGR, but as that name may imply, the role that we have in the dynamic omics group really complements the human and population genomics being done by CGR. And really our goal in dynamic omics is to use mass spectrometry-based proteomics and metabolomics to accelerate disease-based drug development. And this is really centered around the idea of supporting target identification and target validation and informing insights into mechanisms of diseases and, of course, the molecules and medicines that we hope to develop with this data. And so a couple key points I want to get across is that we work across all of the different therapeutic areas. So this would include oncology, respiratory, cardiovascular, and renal and metabolic diseases, as well as our vaccine and immunotherapy groups, as well as our rare disease area, Alexion. And we really support, specifically in dynamic omics, the preclinical pipeline. And so this is going from the identification of a drug target, which is oftentimes a protein, and certainly coming from the proteomics data going to usually be the case. And then, of course, there's a variety of different modalities. These can include traditional small molecules, as well as biologics. That would include things like, obviously, antibodies, but also, next-gen biologic therapies, things like cell therapies or antibody drug conjugates or bispecific antibodies. And then, the work we support goes through the target selection and then the lead generation. So this would, of course, be the generating of a molecule against the target, the optimization of that, and then ultimately the candidate drug state where this is proposed to go into a clinical trial, right? And in our group, in order to be able to support such a large organization and the diverse types of biology we're studying, we need to be able to perform a variety of different omics measurements. This would include things such as just discovery proteomics, post-translational modification characterization, immunopeptidomics, metabolomics, lipidomics, and all of the different flavors of those, such as fluxomics, and then kind of clinical multi-omics. So this we would be considering here in the preclinical space, mostly looking at things like biofluids. And it's also really crucial to get across the point that in some cases, we may need to perform multiple of these omics measurements on a single sample type, or in other instances, we may need to be able to perform a single one of these types of analyses across all of these samples for a given study. And so as we kind of think about this at a high level, what are the three key areas we need to optimize? Of course, significant efforts have gone in the laboratory based, which would be the first two pillars of this, which of course is using liquid handling systems to automate our sample preparation, as well as increasingly high throughput and deep proteome coverage, both DDA and DIA acquisitions. And really, it's kind of amazing that we're able to do this at a scale of now being able to measure 60 to 180 samples a day on some of the timsTOF or Orbitrap-based platforms. And then finally, of course, what I hope to get in today is the importance of the data analysis platform and being able to scale this elastically to be able to support the growing depth of proteome coverage and the number of samples that we're able to generate. Currently in our lab with the technology we have, we're measuring around 10,000 proteomes per year, but certainly we would like to scale that even further. And so if we think about this, then traditionally how a project would have been done, or what does a standard proteomics workflow look like in end to end, right? And so we have some sort of experimental description, and this is really just going to be like an Excel spreadsheet or a PowerPoint slide, transferred from a collaborator. There's a laboratory or analytical stage of this where we're going to have some sample processing, and that's going to depend on the type of sample. This may be done kind of ad hoc. And certainly there's lots of scientist intervention in these steps of randomizing run lists, for example, and determining how that data is stored once the mass spec data is acquired. And then once we think about the post-analytical, right, we have a variety of different pipelines that we may run, potentially with inconsistent formats or storage based on the type of mass spec acquisition we've performed, and then manual intervention at kind of both the more downstream data analysis or quality control steps, as well as the dissemination, which of course is one of the key steps to get the insights that we're generating to the biologists to be able to inform project decision making. And so the challenge with this is, though, of course, that if any one of these steps doesn't scale, the whole system will not scale, right? And so we're really only as good as kind of the weakest link in this pipeline. And so then we kind of pose the question, how can we streamline through an end-to-end flexible platform? And so one of the themes that was identified was that there's lots of things that we can predefine in what I'll get to in thinking about metadata. And this would include things like sample information, right? We likely can capture this in a more controlled way than what would just come in the traditional way of like an Excel spreadsheet. There's lots of processing parameters and that could potentially influence the statistical contrasts that would need to be done. And so this really all the way that we think about metadata, right, in proteomics. And so, the challenge with doing this, once you kind of get to the scale of measuring, 10,000 proteomes a year on the diverse types of samples that I've described, is that you can really think traditionally, you have this then essentially a very long list of studies. You have a very long list of assays, which are essentially the files or measurements that were made in those studies. And then you have a long separate list of the way that the analyses were done, right? And so we think about this as like being lists of lists of lists, right? And so this becomes quite difficult to manage. And so in order to have more of a truly end-to-end workflow that scales, we feel that we need to be able to properly define these inputs and outputs, as I've mentioned. And this needs to be taking into consideration the complex study designs that we have and the diverse data types we're working with. And so we identified a need in the team for a flexible model that enables both repeatable analysis and inter-experiment comparisons. And so what we've really opted here for then is more of a graph-based model using controlled vocabularies, as opposed to maybe a more traditional tabular format or approach to handling this metadata. And so another key point of this is that this also allows for multi-omics metadata integration, right? Since in many of these studies, we may be performing multiple types of omics on these samples. And this is important to highlight that this is possible because, of course, this key core metadata stays consistent across the different models. And so that this is essentially the highlight here would be this core metadata model of defining the subjects, the biospecimens, and the assays, which then allows you to have this study variable that can then, of course, inform the contrasts and the analyses with the input of additional workflow parameters to essentially close the loop and define the experiment that needs to be completed. And as someone that manages a mass spectrometry team of people running these instruments, one of the key aspects of this is that this metadata model allows event-based processing. So every event in this utilizes the metadata in some way. And so, right, there's enormous time savings and removes errors because we can have automatic run list generation from this metadata. And this, as I've mentioned and alluded to, allows for randomization and also the ability to have this across all of our different instrument platforms. And so upon the successful end-to-end completion of the acquisition, right, then this from after the mass spectrometry experiments completed would then trigger the analysis workflow or execution. And then so all of these workflows have a single input of a metadata graph. And so this is important for several reasons. One reason, of course, is that it allows us to be able to quickly reanalyze the data. So in the case of the example, if someone were to remove consent and we were need to be able to repeat that sample, we could rerun the analysis identically without having that sample. And so this means that there's no additional information really then required that's needed to complete the analysis other than the input metadata itself. And so then another important aspect, of course, of doing proteomics in this type of environment is the study-specific QC reports, right? And so we have many different levels of QC, right? Certainly things like system suitability QCs that go on in the lab. But if we think about this more at the data analysis level, right, we can use these metadata models to inform that as well, right? And so, an example of this is, right, looking at things like, hemolysis or platelet contamination or coagulation would certainly be a relevant QC to consider for biofluids like plasma or serum. Something like blood contamination is something that we look to in biospecimens such as tissue, urine, or lavage fluid or CSF. And then certainly when we start to think about, the different types of measurements we're making, phosphoproteomics being an example of this with something like phosphopeptide enrichment efficiency, right? And so, this metadata is also nice because it allows us to essentially generate study-specific QC reports to ensure that, we have the highest quality data that we're reporting to our partners and collaborators. And then, of course, another key theme here that I want to highlight is the importance of being able to share this data and have controlled versioning. And so one of the kind of higher level themes of this is we want to ensure all of the outputs are fair, which I think is a concept that, it was introduced in one of the keynote sessions yesterday, if you're not familiar with this, but this means that we want the data to be findable, accessible, interoperable, and reusable. And that, of course, these results are automatically connected to, controlled vocabularies through these study variables. And so we need to ensure that every step of this analysis is also, versioned and can be fully reproduced. And so, provenance of these workflows needs to be maintained so that we can reanalyze a study also when new technologies, when the people in this room are developing new computational mass spectrometry approaches, right? We can quickly reanalyze those. And so I've mostly talked about this at a fairly theoretical level up until this point, but I do want to kind of highlight, without naming specific mass spec softwares or data analysis softwares that we're using, kind of how we're actually implementing this. And I think that point was a conscious decision because we are fairly agnostic about the tools we use. It really depends on finding the best tools for the biology that we're trying to study. But obviously, there's a large team of system engineers and architects and bioinformaticians that have developed this framework, and it's obviously using a lot of a variety of commercial and open source or open source in-house modified tools. Things like AWS and S3 buckets for data storage. We're able to run a lot of the proteomic searches on the Nextflow workflows, and of course using AWS for like the auto ingestion as well. And this is really then kind of the how we actually implement this kind of end-to-end proteomic strategy. And so then in summary, what I hope I've shown you is kind of how it is crucial for having an end-to-end platform in our group, with considering the diverse type of samples and studies that we're supporting. We've streamlined this metadata capture, and I didn't get to describe exactly how we're doing that, but this really allows us to enable automation, reuse, and visualization across this diverse types of experiments. And of course, the modular development streams in an organization like ours are important for ensuring future improvements as well. We're implementing stringent data quality controls and measures. And then the final point there, of course, being that I think one of the key messages I'm hoping to get across is that it's important to develop both a robust experimental and computational mass spectrometry workflows. And without, having both of those in tandem, we can't really achieve the full goal or potential of proteomics in helping drug development and discovery. And so I would highlight here that I've got the opportunity to talk about this work, but obviously this requires a large team of people with diverse expertise across different groups. And so the kind of two main groups are Sonia Hess's Center for Genomic Research Dynamic Omics group, which is centered around these 4 core technologies. I focused on metabolomics and proteomics as 2 of those, but we also do a lot of flow cytometry and microscopy imaging. And then Sebastian's informatics team he leads, which obviously provides the bioinformatics, but then also the systems architecture and engineering for actually implementing these, because obviously mass spectrometrists can't typically put these platforms or pipelines in place. And then the last thing I would highlight is that both of these groups are hiring. So my team in Gaithersburg, Maryland, just outside of DC, has multiple senior scientist positions for people that I would describe as proteomics scientists. And then Stuart MacArthur's team it has multiple positions for informaticians and bioinformaticians that would be based out of Warsaw, Poland, or Barcelona, Spain. And so please do feel free to reach out and network with us if you want to be a part of this team. So yeah, thanks.
Hi, great talk. So when you say you capture metadata, is it both sample metadata and feature metadata?
Yes, it would be. I mean, from the perspective of today, I mean, I'm certainly more talking about this at the sample level, but yeah, you certainly would, and we'd want to consider feature metadata into this as well.
Right. So and like they both would they be treated the same way?
It's a good question. The feature metadata probably is probably not quite advanced. So I'd probably need to think about exactly how you would differentiate those a little bit. Yeah.
Hi, awesome talk. I love to see an end-to-end process. I think that's something we struggle with a lot with drug discovery. In my lab, we think a lot about target selection. Mm-hmm. So I was just curious about your process and kind of things you've been implementing in the pipeline.
Yeah. That was what I was hoping to avoid having to talk about today, because I as you're probably aware, I'm a little bit limited on exactly what we can disclose there. But what I would say that the key thing to keep consideration is that, right, protein abundance is a great biomarker for, efficacy of any molecule that targets proteins. So certainly we have a fairly sophisticated way at which we kind of are able to rank targets based on protein abundance, right? We don't only look at protein abundance, but what I would say there is you can essentially think about something analogous to the filter that was shown in the first talk today, and thinking about how you go from 10,000 proteins you measure down to maybe a handful of targets, and protein abundance and a variety of different factors to kind of filter that list down.
Yeah, I'd love to talk more.
Yep.